Hello everyone. In this tutorial video, I'll be talking about DNA polymerase III. As you know it very well that for DNA replication, different types of proteins, enzymes are involved, out of which DNA polymerase enzymes plays important role. There are different types of DNA polymerases. DNA polymerase 1, polymerase 2, 3, 4 and 5. Among this, DNA polymerase III is known as heart of DNA replication because the main function of polymerization is carried out by DNA polymerase III. This DNA polymerase III is capable of polymerization in 5 to 3 direction, 5 prime to 3 prime, and it has got the exonuclease activity which is 3 prime to 5 prime, that is used for proofreading activity. This DNA polymerase III, it is known as holoenzyme, which contains almost 10 proteins which are organized into 4 types of subcomplex. I'll be dealing with this each type of subcomplex in detail. The highest polymerization activity shown by DNA polymerase III is 10 raised to 5 nucleotides per minute. It is the true enzyme which is responsible for the elongation process. DNA polymerase III is dimeric structure. It has got the core enzyme. This core enzyme consists of mainly 3 subunits, and these 3 subunits are alpha subunit, which is nothing but the DNA polymerase, epsilon, that is for 3 to 5 proofreading exonuclease, and theta, which stimulate the exonuclease activity. As I said, it is a dimeric, means there are 2 catalytic core, and each core enzyme consists of 3 different subunits: alpha subunit, which is for polymerization; epsilon, which is for proofreading activity, that is 3 to 5 proofreading; and theta, it stimulates the proofreading activity. So this is about the catalytic core, and DNA polymerase III consists of 2 copies of catalytic core. You can see this is the one copy and this is another copy. Now, dimerizing unit, top subunit, this is known as the top. This subunit is responsible to maintain the dimeric structure, means it is responsible for holding of these 2 catalytic core together. Next is processivity units. Here the beta clamp, you can see this beta clamp. This beta clamp is mainly responsible for holding this catalytic core on their template strand, means this processivity of DNA polymerase, it is controlled by this beta clamp. Again, I'll repeat, the holoenzyme 3 is made up of different subunits, and these subunits are classified into main 3 groups. Number 1 is the catalytic core. The catalytic core consists of 3 subunits. Alpha is for polymerization, epsilon is for proofreading, and theta, it stimulates the exonuclease activity. Tau segment maintain the dimeric structure, and beta clamp is responsible for holding the catalytic core on their template strand. And the last one is gamma complex. This is known as the gamma complex. The function of gamma complex is it it places the processivity subunit, that is the beta clamp, on the DNA. Means it helps for loading of beta clamp, and this clamp loader is made up of 5 different subunits. So for loading of this beta clamp on DNA, this clamp loader plays important role, and it requires energy, and that energy is given by the hydrolysis of ATP. The clamp controls the association of core enzyme with DNA. This is nothing but the helicase enzyme, and helicase moves along with the double-stranded DNA. As it moves further, whatever the hydrogen bonds which are present in between these two strands, those are broken, and the single-stranded DNA is made available as a template. Now, as I said, that the helicase is responsible for creation of replication 4. This helicase enzyme is connected to two different DNA polymerase catalytic subunits. So these are two different DNA polymerase catalytic subunit. This is the catalytic subunit which is loaded on the leading strand, whereas which mainly involves in the synthesis of leading strand, whereas this catalytic core is involved in synthesis of lagging strand. Now, the polymerase that synthesizes the leading strand, it moves continuously, and it is the the catalytic core on the leading strand, it is processive. This is because the clamps which is connected to this catalytic core, it helps this catalytic core to remain on the strand for a long period of time. Whereas the clamps which are associated with the catalytic core, which involves in the synthesis of lagging strand, it dissociates at the end of the Okazaki fragment. See, this is let's consider this is the primer, and later on it is extended by the catalytic core, and it continues addition of nucleotide on the lagging strand. When it reached here, now again this catalytic core should get dissociated from the DNA, and therefore the dissociation takes place because of unloading of this clamp from the template strand. So after unloading, after getting dissociated, again this is the beginning of new Okazaki fragment. This template will be pulled over here, so that this site will come over here, this template will come here, and then the primase enzyme will come in the picture. It will synthesizes the primer enzyme. Later on, the primer will get extended by the catalytic core. So again, the reassociation of beta clamp on this template strand takes place, and therefore again because of reassociation of beta clamp with the help of clamp loader, this catalytic core will continue synthesis of new Okazaki fragment till it reach up to here, and this cycle again will continue. The helicase, that is the DNAB, is responsible for interacting with the primase, that is DNAG, to initiate each Okazaki fragment. So the initiation of Okazaki fragment, that is carried out by the primase enzyme. So this is how this all subunit of DNA polymerase III, that is the holoenzyme, plays important role so that the simultaneously synthesis of leading strand as well as lagging strand takes place. So does it explain how the coordination of leading as well as lagging strand synthesis takes place? So this is possible because of all these 10 different subunits which work together. Thank you.